Good afternoon, everybody. I hope you're enjoying your time in San Diego. This time or next time, please come see us. Lineage is located about 10 minutes up the road. I'll refer you first to our safe harbor clause, as I may be making some forward-looking statements. So Lineage is a growing, emerging regenerative medicine and cell therapy, or I prefer to say cell transplant company. I'm going to walk through very quickly a number of the programs that we have under development. But this is a company that you may not have heard of, but I think there's a reasonable probability that you will hear about us in the years ahead. The company is well positioned to be able to execute on some very ambitious goals that we have, and I aim to share some of those goals with you today. So what do we do? Well, Lineage, I described us as a cell transplant company. So what we do is we start with pluripotent stem cells. We expand them essentially infinitely in number. And then we run them through a differentiation process to manufacture just the specific cell type which we aim to use as the medicine. So we never put a stem cell in a human being. We only and always convert those pluripotent stem cells into some other type of cell, the cell that your body has lost due to aging, disease, or whatever reason. That differentiation process is something that obviously spins out a lot of intellectual property, a lot of value for us, and then we use those cells in a transplant, a permanent transplant into the body to a number of different indications. One of the advantages that we have is the starting material that we utilize is the same forever. We go 50 years with a program and we would never change the cell line. It is a permanent cell line. There is no sourcing of new material. We never have regulatory issues changing our donors. And we never manipulate the DNA. There's no gene editing involved. We are following nature's own normal developmental biology process in order to manufacture the different cell types that we work with. This is a pipeline of what we have. 3 clinical programs today. The most notable one I'll spend the most amount of time on is in ophthalmology. And I'll get to that in just a moment. But the facility that we have is a team of about 50 people. We've got multiple GMP suites. And again, this is the horsepower of this company. This is where the differentiation protocols are established. This is where the clinical trial material is manufactured. This is where we're able to take research grade processes and turn them into commercially viable, commercial scale, commercial production, commercial purity and reproducibility and control, all in our own hands, not thrown over the wall, and see what comes back. So Orphan is the name that we have for our lead program. This is where we manufacture replacement retina cells. This is a notable program because it was part of certainly one of the largest cell therapy license deals with our partner Roche and Genentech. This is a deal that came with $50 million up front. It had another $620 million of additional milestones. It had tiered royalties, double-digit royalties. Genentech is going to take on future development. We're finishing the trial that we ran, and we're manufacturing material, but we are also doing tech transfer to Roche and Genentech. So this has been a really exciting validation of our approach. All of us in this room know that cell therapy has been around in some form or another for a very long time, but it is starting to mature. And I think deals like this one and some of the acquisitions that we've seen in the space are illustrative and informative about the future of this branch of medicine. Dry AMD is a problem wherein the retina cells die off. You can imagine just a spot of them, a few of them die off, and it expands outward. We're in San Diego. We know what wildfires are all about. That's the same thing. It's like a wildfire in the back of your eye, and it robs you of your vision. If you could manufacture replacement RPE cells and transplant them safely and durably to the back of the eye, if they are functional, if they are permanent, if they are not rejected, they may be able to provide the function of the RPE cells which you lost. And that's exactly the observation that we have had and enjoyed in the clinical testing of this program. We have been able to manufacture and deliver typically 100,000 RPE cells in a suspension. They're not on a scaffold or anything like that, to the back of the eye, and we've been able to see quite compelling clinical results. The most exciting and really notable clinical results has been the observation that the structure of the retina, or the area of atrophy, this is like an aerial photo. We've colored the area of atrophy, the defined perimeter color there in blue. There's a couple other smaller spots in yellow and red. We have been able to show that by transplanting allogeneic RPE cells, we can make this area of atrophy smaller. Now, if you follow the space in dry AMD, there's a lot of excitement and a lot of money floating around, and people are very keen because some companies have been able to use small molecules to slow the progression of what everyone calls an irreversible disease. It's not an irreversible disease, okay? You can make an area of atrophy actually smaller. And if you do high-resolution OCT imaging, you can actually see all the layers of the retina that have this very clear, visible restoration around the penumbra or the fringe area where the photoreceptors hadn't fully died off. So that is an amazing anatomical, irrefutable, right? This isn't a pain score, right? We're talking about anatomical images that do not change over time, and no human being has the ability to regenerate their retina, right? We're not starfish. So this is from the intervention. We've got now 5 cases of this finding from our phase 1/2A clinical study. And those 5 patients, by the way, have enjoyed an average visual acuity improvement of 12.8 letters on a standard ETDRS visual acuity scale. So that is functional and anatomical data. So we aim to, through our partnership with Roche and Genentech, really change the terminology of dry AMD, erase this notion of it being irreversible, and really show the power of cell transplants and cell therapy. There are millions of people just in this country that have this condition. Fortunately, we have moved our production into a 3D bioreactor. We can generate thousands of clinical courses in a small bioreactor. These bioreactors obviously are much more well controlled than growing cells in 2-dimensional space. So the scale and volume and pricing, our cost of goods gets driven down tremendously. There's no technical hurdle or obstacle that I'm aware of that prevents us from manufacturing in much larger bioreactors and being able to have abundant supply, abundant commercial supply. And we also developed and patented a new formulation wherein this is just an immediate thaw and inject. So these older ideas of having cells that need to be washed and plated and counted and recounted and then eventually put into the patient, we have cells that come out of a frozen state. 5 minutes later, they're going into the injection needle and into the patient. It's a 30-minute one-time surgery, all the benefits of gene therapy, but without the gene editing risks and without having to find just that one patient that has that one particular mutation. I don't care, frankly, why the patient has dry AMD. You could have 1,000 different things going wrong, and probably 1,000 different things do go wrong by the time a cell dies off. It doesn't matter if you replace the cell. You cover everything. This, I believe, is the power and the future of cell therapy. Drop to the bottom. In certain settings, replacing whole cells might provide restorative benefits beyond the reach of traditional approaches. That's not to say there's not a place for small molecules and antibodies. Clearly there are. But in some settings, cells might be better. All right, so here's one of the settings that I think is really exciting, spinal cord injury, another place which has been talked about in cell therapy for a very long time. This can be another opportunity for cells to outperform. What we do here is not manufacture retina cells, but we manufacture oligodendrocyte progenitor cells. These are the cells providing the myelin sheath, the insulation for the nerves. You all remember, probably many of you rather remember, nerves that wire together, fire together. Well, they need to be insulated, so that's what we do. We're providing the cells directly into the area of injury in order to provide an opportunity for restorative benefits. This is very similar to what we're doing with OpRegen and dry AMD, but of course we have a different cell type. This is a program with RMAT designation, Orphan Drug Designation. It's been one of the most important, I believe, speaking for CIRM, one of the most important programs from their early days. We've enjoyed tremendous support from that organization for this program. And it's not a spinal cord injury program. It's a program for demyelinating conditions. We just happen to be focused initially in spinal cord injury. This is clinical data. There are 5 thoracic patients that have now 10 years of data. Nobody's ever had a cell-related complication. Nobody's ever rejected the cells. These patients, you do an MRI, the cells are still there. This data comes from 25 cervical patients. So cervical is really where we're excited because the cervical nerves, when they get injured, that's what wires out to your upper extremities. So if I want to advance a slide, or if I want to drink my coffee cup, or if I want to control my wheelchair, I need upper extremity mobility. So I know we're all excited about people throwing away wheelchairs, and we remember the commercials of people doing that, but we've got nothing in spinal cord injury. There's nothing approved right now. So this is an exciting place to start. We're trying to bring back higher quality of life for individuals by giving them greater upper extremity mobility so they can text and control their lives and dress and all the things that you and I take for granted. The data is very encouraging. From these 25 patients, all but one of them had improvement. There is a difference here with spinal cord injury, to be fair, unlike the setting of dry AMD, where nobody spontaneously recovers. That is a progressive condition. Spinal cord injury patients do enjoy some level of benefit. And so you can see that on the left side, 68% of patients would be expected to have some level of benefit. We had 96% of patients that enjoyed a benefit from our study. So this was very supportive for us for continued development. The problem that we had, sorry, Lucas, I'm going to skip you on this time. They only gave me 12 minutes. Safety. 534 AEs recorded in this clinical trial. One was potentially related to OPC1. I love that. I mean, when you think about the magnitude of benefit you're trying to show against a loud background of 68%, if you have this kind of safety profile, I think that's very telling and encouraging that even a little bit of benefit, and when we talk to patients, that's what we hear. Get me a little bit of angle, a little more angle on my finger, and I'll be able to figure out how to pick up that spoon or pick up that coffee cup. So I'm really excited about that. We have been spending a lot of time working on the manufacturing. I said that with the retina program, that was a core competency for the company. It is very much the case. We've been able to take some original material that was used in the clinical program that I just showed you, and we've been able to increase the methods, the testing, the quality, the scale, the reproducibility. I'm kind of hoping the next slide is going to show some impurities. Yes. The material in blue, that's the original cells that drove that clinical result that I showed you a minute ago. These markers down here, they're anonymized. They are related with undesirable cell types, so think of them as being impurities. The orange stuff, that's what we make now. That's our GMP material today. So you can see how clean this material has gotten in our hands, and we've increased the scale up to 20-fold relative to what was used clinically. That's so critical because I feel that one of the challenges in cell therapy is we're so focused on trying to demonstrate a clinical benefit that we're not always thinking about what is necessary for an actual commercial product to reach patients. So we're trying to solve some of those problems through formulation, scale, reproducibility, and purity. And delivery. This is the next step for this program. We're going to demonstrate a smaller, easier to use device. What I love about this device is it attaches directly to the patient. Why is that important? Well, in the original iteration of this program, there was this large gantry or scaffold, and it would hold the needle above the patient. So you had to disconnect the patient from a respirator, get the needle into position in the spinal cord, deliver the cells, and you've got, call it a minute, because the respirator is off. Now we have a system where the needle floats. It's pistoning up with the chest cavity. So you can take 5 or 10 minutes to deliver the cells, which we think is safer and a better way of doing it. It's also way easier to deploy. It's smaller in the surgical field. This is just a much better way of being able to open multiple clinical sites and have an eye toward a commercial product in the future. So never forget about delivery as part of cell therapy. It sometimes gets overlooked. Okay, auditory neuronal progenitors. This is exciting to me because after we had the validation of the Roche deal and after we received a wire for $50 million, it allows us to start doing more things. Auditory neural progenitors share some of the similarities with the retina program in that there's a huge number of individuals affected and they're losing a certain cell type, they're losing the auditory neurons, they're never replaced, they're never regenerated, and you can't solve this with a cochlear implant, right? Like screaming into someone's ear, a person who lacks auditory neurons, it doesn't matter. They've got nothing to carry the signal. So this is a solution that we're excited about, and this is brand new, and this is something I haven't really started talking about. But when you compare with small molecules, right, our ability to take a program like this from scratch and know that the cells that we begin with are guaranteed 100% able to become our target, our auditory neurons, right? When you do a screen for a small molecule and you set up an assay and you screen 5 million compounds, you have no idea if the chemical space in that library is going to lead to a hit. And even if you do get a hit, you got to go through SAR and all this stuff. We are hoping before the end of the year to demonstrate that we can think of a target, start working with cells, and get it into animals in 12 months. What's the ROI on launching a new program that quickly? I think it's great. So I'm really excited about what we're doing with neuronal progenitors, and we're doing something similar with photoreceptors. This is not part of our Roche deal. This is a totally separate program, but there are many conditions like retinitis pigmentosa where you lose the photoreceptors. That's the prominent feature. So if we can manufacture those photoreceptors and deliver them, that could be another exciting way for us to capitalize on our expertise in the visual field and be able to deliver a different cell transplant to a bunch of people who are otherwise at tremendous risk of losing their visual capabilities. Last thing, those first four were all neuro programs. This is our oncology program. Everyone's seen and been very happy about the excitement and performance of CAR-T, and there's tremendous work going on now with NKs. We manufacture dendritic cells, and the reason that we do that is that allogeneic dendritic cells are really representative of many more products in one. This is a whole separate platform in and of itself. If you manufacture billions of dendritic cells, you can introduce different antigens into those dendritic cells and use those to inform and educate a patient's immune system. So we all know about how this works, right? Antigens are presented on MHC proteins, right? We're skipping that first step. We're preloading the antigen that you want. You partner, or you academic, or we at Lineage, we can choose the antigen we want. We pre-package it into millions of dendritic cells and then put that vaccine approach into the patient. And what we see clinically are very high levels of T-cell response specific to the antigen. So we're getting this screaming loud message to the patient's immune system, which in the right setting in oncology, which I don't have time to go into today, does offer some exciting opportunities because having a higher immune response, a CD4 positive, CD8 positive response can be very effective. And we have seen that dendritic cells delivering antigens can lead to approved products. Many of those products are challenged by the fact that they are autologous. What if you could have an off-the-shelf allogeneic version of it? That's exciting to me. So we've done this. We actually have data in AML from an autologous version of it. We are now focused on an allogeneic. We only work with allogeneic programs. We think that's ultimately the future for many settings is an allogeneic approach. But it doesn't have to be limited to oncology. It doesn't have to be limited to a single antigen. You could do multiple antigens. You could do combinations. You could do infectious diseases. There's so many products that we might be able to imagine developing, hopefully preferably in collaboration. We already do have a collaboration with Immunomic Therapeutics, which is really exciting, and they have some data coming later this year. So this represents a really nice growth opportunity for Lineage. Bigger picture, looks like I'm out of time, so I'm going to show you and remind you, you have over 200 cell types in your body. The ones that are colored in here in orange represent just the 5 that we're working on today. Longer term, we got 195 to go. There's a lot of really exciting things that we could do using this approach. So thank you very much. I will just leave by saying that there's a lot of information on our website. And if you're interested, I think there are like 4 or 5 of us around today, and love to meet you and talk more about what you do and tell you more about what we do. Thank you very much, and enjoy the rest of the conference.